Hello students, welcome to EPG Pathshala. I am Rajat Obroy from Vision Medicare Industries, New Delhi. In this lecture, we will discuss about Pharmaceutical Statistics, paper Pharmaceutical Statistics. In this module, we have 5 learning objectives. 1. Some basic terms used in statistics. 2nd. Probability distribution. 3rd. Hypothesis testing and estimation. 4th. Sampling in statistics. 5th. ANOVA and its application. We will start with the introduction to the pharmaceutical statistics. Pharmaceutical statistics is a crucial tool to design, analyze and interpret an experiment. Rules and regulations have also been promulgated by the US FDA, which makes statistical techniques an indispensable part of fulfilling its recommendation. Good laboratory practice (GLP) and good manufacturing practices (GMP) also necessitates the use of statistics for carrying out the research and manufacturing operation with the highest level of precision. Statistics is an essential requirement in the field of pharmacy with application in testing and quality control, process and analytical method validation, stability testing of the pharmaceutical product, product designing for preclinical and clinical trials, data analysis and several others. Categories of statistical techniques have been developed for the pharmaceutical experimentation, which are A. Descriptive statistics, B. Hypothesis testing, C. Estimation, D. Experimental design. Now we will start with the first learning objective of the module, some basic terms in statistics. Under that, we will start with arithmetic mean. Mean is the most common measure of the central nature of the data, thus can be referred as the center of distribution of data. Mean can be expressed with the equation that you can see on your screen. Second term is median. Median is another measure of centrality and is defined as the value that splits the distribution into half. Median is the central value or can be mean of two middle values if the distribution of the data is perfectly symmetric. Third is the range. Range is one of the ways to express the distribution of measurement and is defined as the difference between the greatest and the smallest value in the set of data. Range calculates only two measurements from a set of data, thus is not an appropriate estimate to describe variation in data. Fourth term is variance. Variance can be defined as the mean of sum of squares and is denoted as shown in the figure with the help of the equation. Fifth term is standard deviation. Standard deviation is the most common measure of dispersion of data and is expressed as square root of variance. It is general estimate of the variability of the population. Sixth standard term used is coefficient of variation. It is a statistical term to measure relative variation in data and is expressed as with the help of an equation that you can see on your screen. Coefficient of variance allows to directly compare the data sets of different magnitude. Its value depend on the nature of data being estimated. A low coefficient of variance may be expected in pharmaceutical analytical experimentation as the variability may be minimal, while a high coefficient of variance is expected in biological experiments. Next term is degree of freedom. Degree of freedom is represented as n minus 1 number of observations minus 1. Next term is accuracy and precision. The terms are used to depict the nature of data and data variability. Accuracy is the measure of how close is the estimated value to the true value. Precision describes the dispersion or variability of a set of measurements. Low standard deviation indicates high precision. The second learning objective is probability distribution. Probability is related to statistics. Probability in a mathematical aspect can be defined as the likelihood of occurrence of event. It may also be referred to the proportion of times a value or a range of value is observed after many observations. Observations are of two types, discrete and continuous. Discrete observations are the ones which are countable finite number, while continuous are the ones that are measured more and more precisely depending on the sensitivity of measuring instrument. Probability distribution is a frequency distribution obtained from a large number of observations. These determine the relationship between the particular variables and the probability of their occurrence. If the variable is discrete, the relationship is called as binomial probability distribution, and if the variable is continuous, it is referred to as normal probability distribution. Now, continuing further, we will discuss about normal or continuous probability distribution. The continuous probability distribution is the one in which the variables are continuous in nature, that is, there may be infinite number of outcomes. Thus, the probability cannot be assigned to the exact value of variable. The normal distribution, also called as Gaussian distribution, is the most crucial probability distribution applicable in many pharmaceutical operations such as concentration of API, active pharmaceutical ingredient in the dosage form, tablets weights in the batch, and many others. It is based on central limit theorem, which states that if a large sample size is removed from a distribution with a finite mean and variance, the distribution of the data is said to be normal. The central limit theorem is based on concept of sample size. If the distribution is originally normal, then the sample size does not have importance, while if the data is skewed, a large number of samples will be required to obtain a normal distribution. Curves for the normal distribution are depicted in the figure as you are seeing on your screen. They consist of two parameters: mean, which is a measure of location, standard deviation, which is a measure of spread. The same can be seen on the screen. The properties of a normal curves are, they are symmetrical about the central value. Second, they are bell shaped, which illustrates that the values in the distribution are near to the mean and the values are far away from rest prevalent. Thirdly, they extend from infinity to infinity, that is minus to plus infinity. Fourthly, the mean, median and mode are numerically equal. Mathematically, the normal distribution can be described using the following formula that you are seeing on your screen. Another important consideration in normal distribution is area under curve. The area between any 2 points represents the probability of observing a value between these 2 points. Since theoretically normal curve comprises of infinite number of values, hence the probability of obtaining any single value is 0. But for any statistical procedure, a probability value needs to be computed in some interval. Thus, to avoid this problem, the concept of standard normal distribution, which possess mean as 0 and standard deviation as 1, is employed. Z transformation approach is applied, which changes the normal curve to standard normal curve. A single table is generated giving areas between minus infinity and z, which can be used to obtain area between any 2 points for any normal curve. Now we will discuss about discrete or binomial probability distribution. Binomial distribution is based on dichotomous data. The event has one of the two possible outcomes. It should be independent, which means the occurrence of one event should not influence the succeeding events. Such a distribution is observed in the following situations. A new dosage form can produce side effects for which the outcome can be positive or negative. B, a new active pharmaceutical ingredient may be efficacious or non-efficacious. C, drug treatment can improve or worsen the disease condition. D, a dosage unit in a quality control test can either be accepted or rejected. It consists of two parameters, that is called as P is the probability of either of the two possible outcomes, and N the number of observations. It can also be presented in terms of mean and standard deviation. Mean is the true probability of success. Standard deviation can be calculated as shown in the screen. A direct effect of sample size is observed on the shape of binomial distribution. As the larger the sample size, the more is the distribution closer to the normal distribution. It is theoretically calculated, and probability of the success of an outcome is known as true mean. The observed values obtained after experimentation are the estimates of true mean. Thirdly, now we will discuss about T distribution. It is called as student's T test. T distribution is most commonly used for the analysis of small sample size. The sample of size n are taken from the normal distribution repeatedly and statistics are computed. The above mentioned ratio in the student's t distribution where n minus 1 depicts degree of freedom, the mean of n samples, the mean of population distribution out of which the samples were selected. S depicts the standard deviation as you have seen on your screen. The curve of t distribution is similar to that of a normal curve, but with more spread and greater area, with the smallest and the largest value at the extremities. The spread in a t distribution is related to the degrees of freedom, as the increase in degrees of freedom result in a tighter distribution with lesser spread. It is better used to determine the probabilities when the S is not known and is estimated from the sample. Fourthly, we will discuss chi-square distribution. This is defined as the total of the squares of the independent normal variables, with each having standard deviation of 0 and S of 1. As you are seeing on your screen, the shape of chi-square distribution is mainly affected by one parameter, number of degrees of freedom. It is mainly applied in the statistical distributions where comparison of proportions and counts are involved. Fifthly and lastly, F distribution. F distribution is obtained from the sampling distribution of the ratio of two independent variance estimates acquired from the normal distribution. The same is also shown on the screen. It depends on two parameters, degrees of freedom associated with each samples and termed as numerator and denominator. It is utilized for testing the quality of two variances and the multiple hypothesis testing, that is called as ANOVA, which we will further be detailing in the same module. Now we will be moving on to the third objective of the module, hypothesis testing and estimation for statistical significance. Hypothesis testing establishes assumptions concerning likelihood of an outcome from the statistical point of view, and further the validity of the assumption is examined using an appropriate experimental procedure. For instance, a well controlled manufacturing operation for a particular finished dosage form has been in use since a long time, has been validated according to US FDA guidelines and produces products of the requisite specifications. Based on this, the assumption is made that the products produced will pass the quality control test. However, the batch obtained does not conform to the required specifications. Thus, the assumption was found to be incorrect and the alternative proposal was made. This explains the basis of hypothesis testing. The hypothesis testing can be applied in situations such as examining the differences in the treatment effects concerning the new drugs and dosage forms to be submitted for government approval, along with the results from clinical trials and the bioavailability studies. If the sample estimate is found to be close to the hypothesized value, the hypothesis is accepted. However, if it is sufficiently far, the hypothesis is rejected, which means a statistically significant variation exists between the hypothesized value and the true value obtained from the sample. The decision to accept or reject the hypothesis is based on the probabilities obtained from the t distribution. As discussed earlier, the following steps can be followed in a hypothesis testing procedure involving a comparison of sample mean and hypothesized mean. Step 1. Define the hypothetical mean against which the sample data mean is to be compared. As you can see on your screen, this is called as null hypothesis. Step 2. Fix the alternative for the null hypothesis to be used. Step 3. Set the level of significance, that is, as you can see on your screen with the example, the significance level, if it is at 5% level, if p is less than 0.05, then is the alpha error. It is thought of as the producer's risk of manufacturer's risk. Step 4. Choose the appropriate sample and calculate the mean and the standard deviation. Step 5. Determine the t statistics so as to make a decision to accept or reject the hypothesis. If t is small, accept the hypothesis. If t is large, implies is considerably different from. To accept or reject this hypothesis, the t value obtained is compared to the value of specified under t table with n minus 1 degrees of freedom at the stated error of 5%. If the table value is lesser than the absolute t value, the difference is termed to be statistically significant. The significance implies that if null hypothesis is true, the chances of observing t-value similar to or greater than the observed one is less than A. We will further discuss about one-sided test and confidence interval. One-sided test, these are utilized when null hypothesis is rejected only for the unidirectional differences of observed means. Utmost care should be taken when selecting the one paired or the two paired test, as the incorrect choice may result in the wrong interpretation of the analysis. For instance, during the testing of new active pharmaceutical ingredient, the null hypothesis may be rejected only if positive effect is produced. Thus, if a negative effect also needs to be evaluated, a two-sided test is used. Further, what are confidence intervals that is used in the statistical estimation? Confidence interval can be obtained for a mean or for the difference between two means as shown in the equation, where D is the mean value or difference of mean. Confidence interval is a better way of expressing results for a bioavailability study. It provides the upper and lower limit for the purpose of comparison, which allows user to identify the equivalency of two products. Confidence limit for the binomial data can be set as follows. In the equation, Z depends on the degree of confidence, P and N defines width of the confidence interval and is also independent of the size of batch. Now we move on to the fourth learning objective, that is sampling in statistics. Under sampling in statistics, first we will discuss about choosing samples for statistical analysis. Samples refer to small number of observations collected from a relatively large population. In approximately all statistical procedures, the small batch size is examined and the inferences are made for the large population based on those small sample analysis. Thus, the choice of selected samples is of utmost importance as they serve as the representative of the whole lot of population. The methods for choosing samples can be categorized into two: probability sampling and non-probability sampling, also known as authoritative sampling. This type of probability samples are depicted in figure 2 that you are seeing on your screen, with their properties provided in table 1 as is also shown in the screen. Table 2 shows the types of probability samples. So there are four types: random sample, stratified sample, systemic sample, cluster sample. As you can see in the table, random sample, each of all the possible units for an experiment have the chance of being included equally in the analysis. Stratified sample, the sample population is categorized into subsets or strata and the samples are selected randomly from each strata. Systemic sample, every nth sample in a systemic sampling is chosen for the analysis, which ensures regular sampling throughout the experimental. Fourth, cluster sample, the sample population is divided into cluster or groups, each containing a subunit. These are the different types of probability sampling as also shown in the figure. Non-probability samples are most often associated with the bias, which may not be apparent and thus not likely to apply the typical statistical method for the estimation of population. They can be used if the probability sampling methods for the particular analysis is either too difficult or too costlier to execute. The sampling plan for the attributes are described in a well-framed document stated as MIL STD 105D. The document specifies the sampling plan in detail with information about the number of samples to be inspected, the criteria for rejection or acceptance. After understanding on choosing sample, now we will discuss about sample size. The sample size can be calculated based on the specification of four parameters. First is the variance. Second is the level of significance, the risk of incorrectability declaring significance and thus rejecting null hypothesis. Thirdly, beta, the risk of incorrectly accepting null hypothesis. Fourth is D, the difference to be detected. The following formula can be used for calculation of large sample size as you are seeing on your screen. Thirdly, we have power. It is defined as the ability of the test to detect the differences if it exists. The following formula that you are seeing on your screen can be used to calculate the power. As a last learning objective of this module, in this we will study analysis of variance, also called as ANOVA. ANOVA is inextricably connected to experimental design. Experiments that are conceived to compare, estimate and test such effects as drug treatments, formulation differences and analytical methods can be designed to yield an optimal return for effort expanded. The experimental results may then be analyzed by ANOVA techniques. A good experiment speaks for itself. The conclusions are often obvious without complicated mathematical treatment. With sophisticated calculators and computers readily available, however, most experiments, if designed properly, can be easily analyzed. In a poorly designed experiment, on the other hand, more than one factor may contribute to an experimental result with no way of untangling the effects of two factors. This is called as confounding. Examples of obvious confounding are: 1. A clinical study in which patients are allowed to take various concomitant drugs other than the test drugs that affect the condition being treated. Second, a comparison of a new tablet formulation to the former formulation for dissolution with the tablets prepared on two different tablet presses, one formulation on each press. Differences in the performance of the presses, for example like pressure, can contribute to differences in dissolution in addition to differences due to formulation changes. Analysis of variance separates the total variation in the data into two parts, each of which represents variation caused by factors imposed on the experiment. A properly designed experiment allows a clear, unconfounded estimate of such variation, or at least can identify the confounding factors if present. Consider an experiment to assess the effects of lubricating agent and disintegrating agent on the dissolution of a tablet. The final analysis of variance would separate the effects of these factors by computing that part of the total variation attributable to the lubricating and disintegration agents isolated from the variation due to experimental error. This separation serves as a basis for testing statistical hypotheses. In ANOVA, now we will discuss about one-way ANOVA. One-way ANOVA can be considered an extension of the independent two samples t-test to more than two groups. In the t-test, the following statistics compares the differences of two sample means to the standard deviation of the differences. As you can see, the pooled variance depends on the variability within each of the two groups. If the two means come from some normal distribution, the difference between these two means, suitably weighted, should also be the measure of variation of the data. A comparison of the estimate of S square from the difference of the two or more means to be within group variability, which can be also called as S square pooled, is a measure of differences between means. If these two estimates of variation are similar, one may conclude that the mean of the group do not differ, that is, the difference between the means can be accounted for the ordinary variability. If the variability due to the difference between mean is significantly larger than within the groups, one can conclude that the means of the groups differ. A large variability of the means is associated with large differences between the means. Since, as noted previously, the variation of means is mean square divided by n, where n is the sample size. The sample variance of the mean is weighted by n to obtain an estimate of mean square. Thus, a statistic that can be used to test treatment or group differences is formed by the following ratio as shown in the screen. The distribution of this F ratio under the hypothesis that the group means are equal is the same F probability distribution previously mentioned when testing for the equability of the variance. The F distribution has V1 and V2 degrees of freedom, where V1 is the degree of freedom associated with the numerator mentioned as G minus 1 and V2 is the degree of freedom associated with denominator equal to summation of n minus G. For two groups, analysis of variance results in exactly the same probability level as the two sample t test. Next, we will see how the multiple comparison helps us. With more than two treatments, if the F test is significant in the analysis of variance, one must determine which of the treatments differ. if a separate test is done comparing each pair of treatments, the chances of finding significance when the treatments are really identical are greater than the indicated by square level. If the square level is 0.05, by definition, one time in 20-year difference will be found to be significant when the treatments are truly identical. If more than one pair of treatments are tested for significance in the same experiment, significant differences are found in more than 5% of such experiments if treatments are truly identical. The concept may be better understood if one thinks of a large experiment in which 20 independent comparisons are to be made at the 5% level. On the average, one significant difference would be expected in each experiment of this kind if the hypothesis of equal treatment means is true. The problem of multiple comparison is complex and many solutions have been proposed. The simplest method of dealing with multiple comparison gives more significant results than would be expected from the ANOVA level. A test is constructed the differences between means using the degrees of freedom and mean square error from the ANOVA. If the sample sizes are the same in age group, a single least significant difference, that is referred as LSD, can be constructed with the help of the equation that you are seeing on the screen, where T is the tabled value of T with the appropriate df at the level of significance. Any difference exceeding least significant differences, as read as LSD, can be considered to be significant. The LSD test should be used only if the F test from the ANOVA is significant. Now we will further study about two-way ANOVA. The two-way model is an extension of the paired t-test in which more than two groups or treatments are compared. As in the paired t-test, each individual, often referred to as block, is subjected to every treatment. Sometimes this model is described as a design in which each individual acts as his own control. In general, the order in which treatments are assigned to individuals is randomized unless a special design such as a crossover is used. A table of random numbers can be used to randomize the order of treatments to be tested on each individual or experimental unit. The analysis of two-way design is similar to the one-way ANOVA, except that an additional source of variation is present, that due to differences among two blocks. For example, if 3 assay methods are to be compared on 6 batches of granular material, the variation due to blocks or the batches is associated with the differences in concentration of active material in the 6 batches. So now we are reaching to the end of the module, and the last thing we will discuss about is bioavailability and crossover designs. Evaluation and analysis of bioavailability and pharmacokinetic parameters are important considerations for the pharmaceutical scientist. Usually, a finished dosage form is compared to some preliminary formulation or marketed formulation regarding relative absorption. The usual method of comparison consists of a clinical study in normal volunteers in which single doses of experiment and comparative formulation are taken in a crossover design. In this design, half of the subjects are randomly chosen to take either one or the other of two formulations on the first experimental occasion, also known as period, leg or visit, and the remaining formulation on the second occasion. A sufficient period of time should intervene between the two periods so that all of the drug is eliminated before the second dose is administered. It is important that power considerations to be taken into account when determining sample size. The FDA recommends that the experiment be of sufficiently sensitive to have 80% power to detect difference of 20% or more between formulations at the 5% level. Usually, 12 to 24 subjects satisfy these conditions using a crossover design. Various parameters can be considered in the comparison of formulas for bioavailability, including 1. Total amount of drug absorbed from area under the blood level versus time of curve, for example. Second, peak blood level, and thirdly, time to peak. Analysis of the results at each plasma or urine sampling time should be discouraged because the multiplicity of tests and correlation of data at proximal time points lead to confusion regarding the true significance level of such test. Also, interpretation is difficult. Other analysis for bioavailability data have been proposed that may be more relevant depending on the drug under consideration. Although these alternatives analysis have much merit, bioavailability data are usually reduced at present to the comparison of 3 parameters just mentioned. This procedure is acceptable to FDA in most cases. The simplest analysis of bioavailability comparisons uses an analysis of variance for a crossover design. Statistical test of hypothesis may be done separately for each parameter, example, relative absorption, time to peak and peak plasma level. This method involves the easiest computation and interpretation but does not take into account multiple statistical tests and correlation of the parameters. Thus, although a multivariate test may be more appropriate, it may often be difficult to interpret. Some statisticians have suggested that hypothesis testing is not appropriate in bioavailability test because acceptance of the null hypothesis cannot logically lead to conclusion that two formulations are exactly the same. Since two different formulations cannot be identically equivalent, rather confidence interval provides more useful information. For example, if two formulas can be said to be within 25% of each other with a high degree of confidence, the clinician can then decide whether the two formulations are similar from a practical point of view. Thank you.